Hi everyone. Thank you for coming to the webinar today. My name is Charlotte Brill and I am a advisor at the Centre for Environment, Fisheries and Aquaculture Science. I'm here today to help facilitate this session today. So this is the second of three sessions on marine research in Pitcairn Islands, led by Cefas and hosted by the MASTS network. We're really excited to talk to you today about the work that has been done in Pitcairn under the UK government's Blue Belt programme, which was established in 2016 to support UK overseas territories with the protection and sustainable management of their marine environments. The government of Pitcairn Islands has been part of this programme since the very beginning, with their no-take MPA also being designated in 2016, which is one of the largest of its kind in the world. Pitcairn now has a marine science base where research can be conducted in one of the most remote locations on the planet. This second session will focus on research delivered by Cefas and the Pitcairn government team with support from the local Pitcairn community. This includes the ongoing fisheries monitoring programme, focusing on understanding the reproductive cycle of Pitcairn's key fish species, and examples of research conducted during Cefas's scientific expedition, including long-term fish and shark population monitoring and investigating coral reef resilience. The third session will be on the 25th of March at 5 p.m, where we'll be joined live by our colleagues in Pitcairn. This will be an opportunity to ask questions to the Pitcairn team directly about researching in Pitcairn, booking and using the Marine Science Base, and any other travel or logistical topics. Once all three sessions are completed, we will be sending out feedback form to capture your thoughts about these sessions and about researching in Pitcairn. Please do fill this in if you can. The presentation today will be recorded and shared to the MASTS YouTube channel, but the discussion section will not be recorded. Questions can be asked at the end of the presentation over the microphone or typed into the chat at any time. This session is scheduled to end at 12 o'clock, but we may slightly run over if there's a couple more questions. I would now like to introduce my colleagues at Cefas, Hannah Wolstenstone and Dr. Valerio Visconti, who will be presenting today. Hannah is a fisheries observer and coral scientist with 2.5 years experience working as the Deputy Lead for Pitcairn under the Blue Belt programme. Hannah has been involved in a number of research projects in Pitcairn, including fish and shark monitoring and coral density research, which she will touch on today. Valerio is a senior fish biologist and leads the otolith science research at Cefas, with over 15 years of experience working with both temperate and tropical fish species. In relation to the overseas territories, Valerio has primarily been involved in projects on life history traits, otolith-based age determination, and otolith chemistry of key fish species, including in Pitcairn, which he will be presenting on today. Now, that's enough from me, so I'll now pass over to Hannah and Valerio for their presentation.
Good morning, everyone. My name's Hannah. I'm going to be presenting the scientific research findings from the last few expeditions to Pitcairn. My colleague Valerio will also be discussing the fisheries monitoring project that is currently ongoing. At Cefas, we work closely with the government of Pitcairn to deliver the Blue Belt programme objectives to support the management of the Pitcairn MPA. So where is Pitcairn? So for those that don't know, Pitcairn is in the South Pacific and is the second most isolated inhabited island in the world. I've added a pin here in the top right where Lowestoft is, which is where the Cefas head office is in Suffolk. So you can see that Pitcairn is almost on the complete opposite side of the world from us. So in 2016, the entire EEZ was designated as an MPA, and to this date is the third largest continuous, highly protected MPA in the world at over 842,000 square kilometres. The only fishing allowed in these waters is subsistence fishing by the local community. With a population of around 40 people, these impacts are negligible. And you can see the MPA is shown on this map here, and the hatched areas are the coastal conservation areas where they are allowed to be fishing. The UK government's Blue Belt programme has been running since 2016 and it works across 10 overseas territories, with the Pitcairn MPA being one of the first to join the programme. There are 5 main themes to the MPA management plan, covering governance and management frameworks, understanding and protecting biodiversity, managing human impacts, compliance and enforcement, and capacity building and ocean literacy. Pitcairn Islands Marine Monitoring and Research Plan is designed to focus interested scientists and stakeholders to collect, analyse and interpret information which is beneficial to the success of the MPA. Plan is organised into themes that align with the objectives of the MPA and outlines the research and evidence gaps of particular importance to the Government of Pitcairn Islands and will be reviewed again later this year. This first monitoring plan has an emphasis on establishing the baseline conditions and putting the plans and processes in place to ensure future effective monitoring of the MPA. In order to conduct baseline assessments understanding the local subsistence fishing species, a fisheries monitoring programme has been established. So I'll now pass it on to Valerio to provide more on this.
All right, thank you very much, Hannah and Charlotte. I'm Valerio, and as has been said before, I've been looking to that fishery monitoring plan in the last few years since we started working on this in 2021 planning. And what I'm going to show you today are some of the preliminary results on the life history, traits, and reproductive biology of the key fishery species from Pitcairn Islands. When we started working on the plan, we were sure that we were looking at something that was under fishing pressure, which was not at the same level that we see, for example, the North Sea or around the English islands. So the reason why it's still important to look at these species is by looking at natural population dynamic baseline. We think because these populations are not easily exploited, if at all, that will give us a very good overview of the fish biology insight and for an unexploited population. So we then can think about looking at the resilience and recovery of these species, just because some of those target species at Pitcairn, they have some vulnerability perhaps around the reproductive strategy and biology. So, for example, some of the groupers can go to spawning aggregation, and that these can be easily targeted by fishermen. So it will be interesting to study these as well. So that will give us more information about that. And then in the kind of a perspective of ecosystem-based fishery management, we want to have those life histories, traits coming from the Pitcairn to really understand and inform the ecosystem roles and potential look at some environmental change responses to that. So just a couple of ideas behind the investigation of these species. So in 2022, we decided, according to the locals, to start working on 3 main species with the biological data collection, and the feedback that were coming from the locals were they were interested in to develop some study on these 3 species that I'm showing in this slide. So one is the yellow-hedge littertail Variola luti. The other one is the black-tip grouper Epinephelus fasciatus red snapper. And another one is the non-we, as they call locally, is the Bermuda sea chub Kyphosus sectatrix. So what we did in 2023, in February, we get there with the expedition, with the silver supporter. We delivered the training. We updated some of the forms and the logbooks. We took a look of how to dissect the fish, how to take the normal parameters, and we started the collection of the data, actually. That was very, very efficient, and it really proved to be a very good way of delivering the training and getting the locals involved into this process. So the 3 main, tasks of the biological data collection involve the measurement of the fish. So the fish morphometrics were taken, so length, weight, and so on. Then we focused the training delivery on the gonads extraction to look at both the macroscopic and microscopic aspect of the gonads, and then infer into the reproductive biology and cycle of the fish. And then we move into the otolith extraction from the fish. If you know, very familiar with these, so the otoliths are actually the ear bones of the fish, and they are used for giving more balance in the water and understanding of the surrounding for the fish in the environment. So we extract them and we use them to calculate the age, and I'll show you some slides later. So with this in mind, we started the collection, as I say, in 2023, and these tables show you a bit of what we have achieved so far in terms of data collection. In the first expedition, obviously, we had the chance to go to Henderson Island, Oino, and some other places like 40 Miles to collect fish over there. So far, we have collected 715 fish, which is, say, a good achievement for what it is. Obviously, we had some up and down in terms of data collection due to adverse weather or other commitment of the people in the island. But so far, so good. We have a good, bunch of data that we are starting working on. So the results, the preliminary results I'm going to show you today are related to the first batch of samples that we collected, approximately 400 fish. So the first task we approached was to look at the reproductive biology of the fish with the samples that we had in-house until last year. So we made some processing on the both microscopic and microscopic stages by working on the histology of the gonads. And the first task that we kind of developed was trying to understand if the sex of each fish and species was assigned correctly. And we've done this by looking at the histology, as I said. Surprisingly, the 49% of the Epinephelus fasciatus, so the red snapper, was misidentified in terms of sex. So either was assigned as a male instead of a female or vice versa. This didn't happen for the Variola lutea and Kyphosus, possibly because they have a different kind of a structure in the gonads, but that was a very good start point to say, okay, we're going to create this table to help the data collection and actually assign in the first instance the right sex of the fish. The next step was to look at more kind of the sex ratio and the sexual ontogeny of the species there, and we found out that there was a kind of some bias in the data collected with some more male biased presence of for the Pinephelus fasciata, for example. The Variola lutea was much more biased on the female side, but overall the Kyphosus, for example, was very balanced in terms of number of samples between male and females. On the other hand, we looked at the sexual ontogeny for the groupers, because we know for many species of them there is a change in their sex during the life histories, and we got the confirmation that there is a change from female to male sex in both of the Pinephelus fasciatus and the Variola lutea. Nothing really new, but it was very worth to get this confirmation for the local population. And fair enough, but what we actually experienced that during the first 6 months of our sampling, say February to September, October, we couldn't find any fish that had signs of spawning activity, either in the microscopic and microscopic stages, the histology. So in these 6 months, we missed the reproductive, the spawning activity. So we are carrying on with the data collection. We hope to really find the spot and the period of the year where these fish spawn, just because we know that might be spawning aggregation, and is very, very important life history trait that we need to kind of investigate. So we move on now into more the work in progress part of the project, is where we start looking at the otoliths, so the structure that I showed you before. Our lab at Cefas in Lowestoft is specialized into processing otoliths. We do process 60,000 otoliths per year for the normal data frame collection for the more industrial fisheries that we have around the English waters. And so these are the otoliths look like for the 3 species, and the image below show you how we actually take picture of all the whole otoliths before processing, because then we apply some very nice AI shape analysis base that I'm going to show you in a minute. We process all of them in-house at Cefas, and For those of you that are not familiar with the technique we use what is called dendrochronology approach. That is the same technique that I use for aging the trees, the normal trees that you have in the woods. So we use the same approach. We section those otoliths that I showed you before. We create a thin transverse section, and we actually look at the rings deposited annually in the otoliths. With that in mind, we do this for all the fish that we collected, at least in those first 6 months, and we obtain the data based on the age, and a little table here to show you what we found so far with these size range. So we found out that Pinophelus fasciatus lives up to 31 years, Variola luti up to 24, the Nanwi, so the Kyphosus, up to 20. These are very preliminary results. We are going to present these more in detail at the Coral Reef Symposium in July. There is more data coming up, so I'm hoping to then generate more fishery based data, like looking at the growth curves and some other biological parameters for them based on the age. As I mentioned before, we take pictures of all the material that comes to the lab. One is taking a picture of the whole otoliths, because by looking at the shape of the otoliths, we can infer about their kind of behavioral differences in growth patterns across islands. So in this slide, I show you what we have done with the 2D shape analysis of two species, so Pinophelus fasciatus and Variola luti, from La Réunion, which is an overseas territories of belonging to France, and the Pitcairn Island. So what we found is that the shape of the otolith is different, and this is because fish at different latitude, different places in the water, they grow in different ways. This is reflected in the otoliths. So the otoliths can be used in that way to, highlight and understanding the population structure, the adaptation in the coral reef, but as well, more into the fishery side, we can use them to identify stocks. So this was a little work that we developed with Infirmerie in France, just to understand if it was possible to use the otolith shape of these two very important species in that way. So again, this is going to be presented at the Coral Reef Symposium in July in Auckland. The last bit of work, which actually came earlier, straight after the 6 months that we sample some of the fish. So we focus on Kyphosus species because some of the literature was reporting the Kyphosus species in the water of Pitcairn Island being the Kyphosus pacificus. However, one of my PhD supervisor, which is an expert in these families, said, look, Valerio, this is not possible, that this is Kyphosus pacificus. Let's get some fin clips out of the samples and investigate further, because I suspect these are different species. And actually he was right. So it turns out that after running some DNA analysis, this is Kyphosus ectatrix. So it's really kind of giving more use of the samples that we collect at Pitcairn. So that was a very nice kind of a discovery there. And that's my last slide. What we doing now is trying to kind of get the local people to keep recording the data, keep fishing as they do, to get more samples to work on and get a better idea with that. As I said before, we are going to present some of these preliminary data at the Coral Symposium, and with that in mind, I'll pass back to Hannah for the rest. Thank you.
Thank you very much, Valerio. I'm now going to cover the other main research findings from expeditions conducted since 2021. The main research findings for these generally fall within Theme 2, Understanding and Protecting Biodiversity, and Theme 3, around managing human impacts from the MPA management plan I mentioned earlier. The first of these expeditions was Operation Farfalla in 2021. It was led by the Pitcairn community and focused on collecting video data to determine fish and coral diversity and abundance and shark diversity and distribution to create baseline data for future monitoring. Additionally, data were also collected on the coral reefs to understand their resilience to climate change. Operation Redfish in 2023 was led by Cefas and focused on repeating the video data collection on fish diversity and abundance, as well as mapping a nearby seamount and collecting brand new video data on the fish diversity and abundance there. In addition, this survey was joined by Edinburgh University, who collected eDNA data. If you joined in the last session, we heard from the Edinburgh University team about this work. Last year, Operation Gannet journeyed to Owino and Henderson, with Cefas's focus on collecting repeat video data on fish and shark diversity and abundance, for long-term monitoring, as well as adding new research on the corals zooxanthellae to investigate how susceptible the corals might be to climate change. Additionally, in 2024, Operation Putu Putu was led by the Natural History Museum and studied the prevalence of plastic pollution ingested by seabirds. Unfortunately, Cefas weren't able to be present on this expedition, so I won't go into any detail here. In 2021, 23, and 25, surveys were conducted using baited remote underwater video systems, or BRUVs for short, and this monitoring technique has been implemented across 13 overseas territories as part of the Global Ocean Wildlife Analysis Network, or GOWAN project, in collaboration with Blue Abacus. The aims were to collect video data to determine fish and coral diversity and abundance. During Operation Farfire, 77 deployments were completed across the 4 islands by the community during COVID. In 2023, Operation Redfish conducted 52 deployments across Henderson, Owino, and a new location, the seamount called 40 Mile Reef. In 2025, Operation Gannet conducted repeat deployment on all locations at Henderson and Owino. The video analysis for these drops, though, is ongoing, so I'll only discuss the 2021 and 2023 results here. So what actually are BRUVs? BRUVs are a setup made up of a mount with stereo camera systems to allow for size quantification and a bait arm, which you can see here, to attract predators. In 2021, these were also modified to downward-facing cameras to conduct drift videos collecting information on benthic habitats and diversity, which will be discussed in a bit more detail later. So the key findings from this work showed that each island has a unique fish assemblage that reflects the variable habitat types found across the marine protected area. We recorded 203 species in the first drops in 2021, and this went up to 220 different species across 2023. We also saw a total of 4 species of shark, which were the grey reef, the Galapagos, the white tip and black tip reef sharks, and these were recorded at 44 to 51% of deployments. At Henderson and Owino, we've seen consistent diversity, abundance, and size distributions between the surveys. And in 2021, we also had humpback whale mother and calves recorded at Owino, Ducie, and Henderson. However, these weren't seen in the 2023 drops, as these second drops were done in February, which is outside of the whale season. So here we can see on the graph, you've got dark blue shows the 2021 data and the light blue shows the 2023 data. These graphs are comparing taxonomic richness, total abundance, and mean fork length for the 2 years across the 5 sites. Henderson and Ducie have the highest taxonomic richness, and Ducie has the highest fish abundance, with Pitcairn having the lowest, but their fish are generally larger than other islands. 40 Mile Reef has a low diversity but had the largest fish of all, which reflects the deeper, more open seamount habitat present, where the seamount top is at about 65 to 70 meters depth. Comparing the surveys, taxonomic richness, abundance, and sizes show good consistency for both Henderson and Owino. We only have comparison for these 2 islands currently, as there's only 2 years worth of data available for them. So our key findings from this work are that fish abundances across the whole MPA are some of the highest seen in subtropical sites across the globe. The success of the first expedition has shown that a mix of modern technology and local enthusiasm allows important research to go ahead even when international travel isn't possible, and the consistency in BRUV's results is helping to build up a baseline for longer-term monitoring. So in 2024, we did a more in-depth study to determine the diversity and distribution of sharks using the data from the 2021 expedition plus information from the literature. Most common species found in the data were the grey reef, the white tip reef, and the Galapagos sharks. Grey and white tips appear to associate most commonly with coral cover, which drives the high numbers at Ducie and Henderson, with Ducie also having higher numbers of smaller sharks, indicating that this might potentially be a nursery area for them. In contrast, the Galapagos sharks seem to be associated with macroalgae cover and are found more predominantly around Pitcairn, where this habitat is much more common. The 11 species seen across all data and literature had 3 that I just mentioned were the most common across both. The sharks are apparently rare around Owino, and the reason for this is currently unclear, but is potentially linked to historical fishing pressure. Further research is required to understand whether there has been an increase in shark population since the MPA was designated and management strategies implemented. This could be done through continuous yearly BRUVs data collection, as we're working on at the minute, but it would also be interesting to do some acoustic tagging to understand the movement of the sharks and how they use the different habitats found within the MPA and investigate any potential island connectivity. We also were in conversation with the IUCN's ISRA branch, and at the end of 2024, 2 new important shark and ray areas were designated, one for Henderson and one for Ducie, indicating that they are a really important area for these sharks. Following the publication of a Cefas paper in November of 2022, which recommended a climate change resilience assessment of Pitcairn's coral reefs, we produced a report aiming to assess the physical and biological factors that affect the ability of coral reefs to resist and recover from climate disturbances. 11 resilience indicators were used to conduct the assessment, including herbivore, biomass and diversity, coral diversity and size distribution, bleaching resistance, macroalgae competition, substrate suitability, and the variability of summer and winter sea surface temperatures. All anthropogenic pressure indicators were also estimated using proxies for nutrients, sedimentation, fishing pressure, and physical human impacts, i.e. anchor damage. To conduct this assessment, we used video data collected in 2021 from both the stationary BRUVs set up for monitoring fish and the modified drift BRUVs for corals and other benthic flora and fauna. We also considered historic thermal stress and future IPCC sea surface temperature projections to indicate historical bleaching events and risk for future bleaching. The study highlighted spatial variability in the relative resilience potential between the islands, with Pitcairn having relatively low or medium to low resilience. Ducie sites were all classed as medium to high, and the majority of Henderson and Owino sites were classed as high or medium-high resilience. The key indicators for future monitoring should include herbivore diversity and biomass, coral diversity, bleaching-resistant taxa, substrate suitability, small colony density, and mature colonies, as these had the greatest influence on the relative resilience rankings. Pitcairn Island has the highest anthropogenic pressure, being the only one that's inhabited, but typically the lowest relative resilience and thus the lower relative priority for additional management action. Despite this, local targeted fish stock should be sustainably managed to avoid overexploitation due to the high dependence on subsistence fishing of the local coastal community. The historic thermal stress records from satellite data suggest that the islands have never experienced a severe warm water bleaching event, and a high percentage of thermally sensitive corals supports the hypothesis that Pitcairn Islands have remained a climate refuge to date. Bleaching events are not predicted to occur until the 2050s or 60s, even under the IPCC high fossil fuel emissions scenario, and much later if global climate policies are implemented. The naive reefs are likely to have low resistance to bleaching and display high mortality at these events, but could exhibit high recovery from future events. Further research is needed to determine the recovery potential for these coral reefs, though. Our recommendations from this work are to maintain and strengthen protection and enforcement of the MPA to ensure it continues as a global reference site and to act as a key research area on the impacts of climate change on resilient reefs. Carrying on from this resilience assessment and seeing from seeing the corals in person in 2023, it became clear that the corals seemed to be quite pale there, and we were concerned about what this might mean for the health of the corals, even with the lack of bleaching records or predictions. In discussion with the local community, this seems to be a natural paleness and not indicative of coral bleaching. So we've started looking into the densities of the zooxanthellae, as well as starting to see what species of them are present. This work is currently ongoing, so I can't present much here just yet, but in 2025, we collected 48 samples across multiple genera of corals from across Henderson and Oeno that are being analysed for the zooxanthellae species. And we collected 15 fragments from one Bacillopora species to assess the densities in zooxanthellae to compare against other corals around the world. In 2024, we also collected 4 fragments for density assessments from Pitcairn Island. Provisional results from this density analysis indicate that the zooxanthellae at the Pitcairn Islands are not less dense than those from corals elsewhere in the world in their natural conditions. So the normal densities we tend to see worldwide in healthy reefs is 1 to 2 times 10 to the 6 cells per centimetre squared, whereas here we're seeing densities between 1.5 times 10 to the 6 and 1.1 times 10 to the 7 cells per centimetre squared, showing a huge variation in densities across the different locations. The results are suggesting that there may be a difference between the inner and outer branches of each colony as well, which you can see in the left-hand graph. All the colonies are very varied though, with where they were located. So Pitcairn samples were taken from a large, deep natural rock pool and are not assessing for intra-colony variation. And you can see that on the left-hand side. So this is a location called St. Paul's Pool, and it's a natural rock formation. From Henderson, we were working from the fringing reef, which you can see in the middle photo. And for Oeno, we took from sites within the inner lagoon and the outer reef crest as well. But at the minute, we only have the results from the inner lagoon colonies. All of these samples were collected between 1 metre and 9 metres depth, and no correlation has currently been seen in the depth in that top 10 metres of water. But we do have to note that the sample sizes are quite small at this stage. Data collection is still ongoing, and the inner and outer branch looking for intra-colony variation is only available for the minute for Henderson and Oeno. So other research that's been done on these expeditions externally to Cefas includes looking at the mesophotic coral reef refugia, so doing similar work to our BRUV's work, but just undated. We also had some work done on the biogeochemical profile of the surface microlayer. That was done last year, so the results for that one are still ongoing as well. Multi-scale mapping of Henderson and Oeno. We have got ongoing work on the seabird population health from the Natural History Museum. There's also been work investigating the humpback whales through identification and other studies. And as I've already mentioned, Edinburgh University have joined us previously to look at water carbonate chemistry and eDNA. So the Marine Science Base at Pitcairn is building to become more capable of a wide variety of marine research. There is a Marine Science Base information pack that can be shared with you guys if you're interested. There are several locals that have also been trained in a few different research techniques, such as the coral fragment collection, fishery sampling for the gonads and otoliths, basic water sampling, BRUV's deployment, ROV piloting and maintenance as well. So the team are very capable, quick learners, and are very engaged in protecting their MPA. This site is also on the World Marine Stations Atlas and will link you to the MSB's website for more information. So in terms of travelling to Pitcairn, some of the difficulties that you see are just the sheer amount of time it takes to get to Pitcairn. So initially, you need to make sure that you can fly yourself out to Tahiti in French Polynesia, and then from there, you have to get an internal flight with Air Tahiti to Mangareva, which is one of the outer islands of French Polynesia. This is where the MV Silver Supporter will meet you and take you all the way to Pitcairn over 2 days. For equipment, This can be either freighted between Pitcairn and New Zealand. This only happens 4 times per year though. Or the other alternative is you can fly with the equipment out, but returning with it can be quite difficult because of the restrictions in cargo drops and that sort of thing to make it onto the flight. So for research logistics, research permits must be applied for and approved by the council. You also have landing fees and berth fees to pay for. The local store is open 3 times a week for food and drink provisions. Local expertise are available for hire at full day or half day rates as well. And we're exploring what options there could be for future research opportunities and funding in the future. It's also important to note that any outer island excursions will require additional logistics and health and safety considerations, which would be discussed and organised with the team on Pitcairn. So that's everything from me. While we have a quick think and formulate some questions for us, if you've got anything, yeah, feel free to drop them in the chat or raise your hand. And while we're doing that, I'm just going to share really quickly the Marine Science Base website for you. So what we can see here is the information about the Marine Science Base, Pitcairn MPA, and then they link to the environment page for the Pitcairn Islands as well. There's some lovely photos that you can see to sort of give you an idea of the quality of the accommodation. The accommodation and the sort of lab space is all integrated into one building as well, so it's all handy and on site. There's a little video that you can watch as well in your own time that was produced by Protect Blue to sort of promote the Pitcairn MPA. And you'll also find here are a number of different documents to read and go through in terms of what equipment's available, what fees there are for using the Marine Science Base, and other agreements, that sort of thing. And then there's a bunch of health and safety details at the bottom. Yeah, that's everything from me.